Hi all, welcome back to Research PhD for All for the first paper discussion. So I wanted to get started with something that has really changed the world during COVID times. That is the COVID vaccine, the mRNA one. Not the idea of the mRNA vaccine, but how the trial was conducted and the efficacy determined. I think this study really had a profound impact on the world because the resulting outcome has literally saved lives, especially for the most vulnerable members of our society, such as the elderly and those with pre-existing conditions. Additionally, this is also the most fastest developed vaccine, by fastest I mean brought to the market, in the entire history of mankind. Now here's a chart that I found about vaccine timelines for past vaccines. I know that vaccines have become a very controversial topic these days, especially the one for COVID. However, if you look at history, vaccines have played a crucial role in saving us from very deadly disease that used to wipe out entire villages at a time, with diseases like chickenpox, polio. Literally every vaccine we get as a kid was a huge, a massive, a ginormous problem at one point in history. We do really take a lot of things about today's life for granted, and that's thanks to vaccines. In this video, I want to talk about the randomized control trial that led us to approving the vaccine. The method is generally similar to other vaccines and medicine in general. Again, I want to note that similar does not mean identical. The point of this video is not necessarily to get you ready to run your own RCT. Those things has to go through a lot of regulations and checks. You need to actually consult with a professional to run an RCT, especially if it's about injecting or feeding anything. The video will help you understand the process we have accepted to generate results that can be used to determine okay or not okay on this vaccine. Another reason why the setup of the RCT is so important is because in many cases, it costs more to run the RCT with thousands of people than to conduct the research to come up with the formula for the medicine. I also want to admit that I won't go into too much detail about the mRNA biology stuff in this video. That may be a topic for our future video. I admit it is an important topic and will serve humanity well in the future, but this video will be limited to the content in the paper. The vaccine was developed by Moderna, at least the one in the paper that I'm talking about today. mRNA is the technology of messenger RNA, and 1273 should be their code word for the compound. The paper is titled Efficacy and Safety of the mRNA 1273 SARS-CoV-2 vaccine. The paper was published in the New England Journal of Medicine on February 2021. The paper has a large number of co-authors, and that just means that it was a joint study between many researchers. And that makes sense for a study of this magnitude. Drs. Baden and Sahly contributed equally to this article. Now, I could have easily discussed his counterpart from Pfizer, BioNTech 162b2, which is similar in many ways actually. I will put a link to the other paper in the description. So let's jump into the paper. The paper discusses the conduct and findings of phase 3 randomized trial. This trial enrolled about 30,420 volunteers in 99 centers across the country, across the US. And I'll come back to the specifics of enrollment, baseline demographics, how vaccines were administered, the methods used, analysis conducted in a little bit. But I wanted to get started with the actual structure of the contents of the paper itself. Now, this structure is something you will find to be similar in many papers. And in a later video, I can talk about how many papers are structured and what's within each section of the structure. The paper is divided into the following sections. It starts with a bit of a background and intro into the issue itself. Then it discusses the methods used to conduct the research, followed by results and statistical analysis. Then the paper concludes with a discussion of results, and we will follow the same order as is in the original paper. Let's start with the intro. The novel coronavirus 2, commonly known as SARS-CoV-2, which stands for, the SARS part stands for Severe Acute Respiratory Syndrome, Coronavirus 2, came into being around December 2019. This is according to the paper. I believe there's some arguments around this when it originated, but I'm going to follow the original paper. I think everyone remembers well about the origins of the virus. In the beginning, many control measures such as masks, physical distancing, testing, contact tracing, and isolation, such as lockdown, were tried to control the spread, but with limited success. We needed something that could help build immunity against it, such as a vaccine. There were attempts to build a vaccine through multiple ways, but mRNA was one of the most flexible and efficient. And that's why we ended up choosing this one. By January 2020, we had determined the genetic sequence of the virus, and mRNA 1273 was developed by Moderna and Vaccine Research Center at the National Institute of Allergy and Infectious Diseases, NIAID, which lies within the National Institute of Health, NIH. Essentially, mRNA 1273 is a lipid nanoparticle, LNP, which means that it's an encapsulated mRNA vaccine expressing the perfusion-stabilized spike glycoprotein. Its predecessor worked well with animals. Also, by the way, this was not the first mRNA vaccine to develop and get published. Before this, the mRNA vaccine developed by Pfizer and BioNTech, commonly called BNT162b2, was also considered to be recently developed. Now let's talk a little bit about the methods used in the trial. But before we jump into what actually was done, let's talk about the timeline and the regulations involved, because there is a lot of regulation involved in this study. The trial lasted between the 27th of July and the 23rd of October in the year 2020. It was conducted according to the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use, Good Clinical Practice Guidelines, and applicable government regulations. All participants signed a written consent form, which should come off as no surprise. The trial investigational new drug sponsor, Moderna, was responsible for the overall trial design, and they did take input from the Biomedical Advanced Research and Development Authority, the NIAID, the COVID-19 Prevention Network, and the trial co-chairs. Moderna was also responsible for site selection and monitoring and data analysis. Investigators are responsible for data collection, but are unaware of participant-level data. Designated team members within Moderna have unblinded access to the data to facilitate interface with regulatory agencies and the data and safety monitoring board. All other trial staff and participants remain unaware of the treatment assignments. The trial was still ongoing at the time of the publication of the paper. Now, let's jump into the design of the study. Basically, the study followed the following design. Participants were at least 18 years of age or older with no known history of COVID infections, with locations or circumstances that does put them at an appreciable risk of infection, a high risk of severe COVID-19, or both. They used the FDA draft guidance to select sites and enroll participants, such as to increase the number of persons of different demographics, including racial and ethnic minorities, in addition to other risk adjustments. The participants were randomly assigned in a 1-to-1 ratio, so half were in treatment or the vaccine and half were in placebo, who also receive a vaccine, but that placebo vaccine only has salt water in it. Assignment was stratified on the basis of age and COVID complications risk criteria into the following risk groups: persons 65 years of age or older, persons younger than 65 years of age who were at a high risk or at risk for severe COVID-19, and persons younger than 65 years of age without high risk or not at risk. So what does being at high risk means? Participants younger than 65 years of age were categorized as having high risk or severe COVID-19 if they had at least one of the following risk categories. And these risk categories are based on the CDC's, Center for Disease Control and Prevention's criteria, available at the time of trial design. This list includes chronic lung disease, such as emphysema, chronic bronchitis, idiopathic pulmonary fibrosis, cystic fibrosis, or moderate to severe asthma, cardiac disease, such as heart failure, congenital coronary artery disease, cardiomyopathies, or pulmonary hypertension, severe obesity, which is calculated based on body mass index, which means if the weight in kilograms divided by the square of height in meters is greater than or equal to 40, diabetes, which can either be type 1, type 2, or gestational, liver disease, or lastly, infection with the human immunodeficiency virus, or the HIV. The mRNA 1273 vaccine, provided as a sterile liquid at a concentration of 0.2 milligrams per milliliter, was administered by injection into the deltoid muscle according to a 2-dose regimen. These injections were given 28 days apart. They were given in the same arm and in a volume of 0.5 milliliter, which contains 100 micrograms of the mRNA 1273 compound, or just a saline placebo. Now, let's talk about what kind of data was collected within the study And by that, I mean besides the baseline demographic characteristics of the participants. Data was also collected for safety assessment purposes and for investigation into efficacy. Let's start with the safety assessment part because a lot of measures were taken to ensure the safety of the participants. Safety assessment included monitoring of solicited local and systemic adverse events for 7 days after each injection. And I'll come back to what these things mean in detail a little later. Unsolicited adverse reactions for 28 days after each injection, adverse events leading to discontinuation from a dose, from participation in the trial or both, and medically attended adverse events and serious adverse events from day 1 to day 759, which means almost 2 years. On to the outcome side. The outcome of the experiment was measured through 2 endpoints. The primary endpoint was the efficacy of the mRNA 1273 vaccine in preventing a first occurrence of symptomatic COVID-19 with onset at least 14 days after the second injection in the per protocol population, which means among the participants that were seronegative or had no COVID at baseline. This was done by an independent assessment committee that was unaware of the group assignment. COVID-19 cases were defined as occurring in participants who had at least 2 of the following symptoms, either fever, which means a temperature above 38 degrees Celsius, chills, myalgia, headache, sore throat, or new olfactory or taste disorder, or as occurring in those who had at least 1 respiratory sign or symptom, including cough, shortness of breath, or clinical or radiographic evidence of pneumonia, and at least 1 nasopharyngeal swab, nasal swab, or saliva sample, or respiratory sample if the participant had to be hospitalized that was positive for the virus from RT-PCR test, where RT-PCR stands for reverse transcriptase polymerase chain reaction. The consistency of vaccine efficacy at the primary endpoint was evaluated across different subgroups, which includes different age groups. If you remember from the age group classification from the past, one age group classification was 18 to under 65 years of age and 65 years of age or older. Age and health risk for severe diseases, which means 18 to under 65 years of age and not at risk, and 18 to under 65 years of age and at risk, and 65 years of age or older. By sex, which means by male or female, by race and different ethnic characteristics, and risk level for severe COVID-19 illness. The study also had a secondary endpoint, which was the efficacy of the mRNA 1273 vaccine in the prevention of severe COVID-19, which was defined by the following criteria: respiratory rate of 30 or more breaths per minute, heart rate at or exceeding 125 beats per minute, oxygen saturation at 93% or less while the participant was breathing, ambient air at sea level, or a ratio of the partial pressure of oxygen to the fraction of inspired oxygen below 300 millimeter of mercury, respiratory failure, acute respiratory distress syndrome, evidence of shock, which means systolic blood pressure of under 90 millimeters of mercury, diastolic blood pressure of under 60 millimeters of mercury, or a need for vasopressors, clinically significant acute renal, hepatic, or neurologic dysfunction, admission to an ICU, or death. An additional secondary endpoint included the efficacy of the vaccine at preventing the virus, the disease, COVID-19, after a single dose, or at preventing COVID-19 according to a secondary or less restricted definition given by the CDC, which is having any symptom of COVID-19 and a positive test according to RT-PCR tests. Let's now jump into the statistical analysis performed in the study. For analysis of the primary endpoint, which was the efficacy of the vaccine in preventing a first occurrence of COVID-19 with onset at least 14 days after the second injection, the trial was designed for the null hypothesis that the efficacy of the vaccine is 30% or less. A total of 151 cases of COVID-19 would provide 90% power to detect a 60% reduction in the hazard rate. That is 60% vaccine efficacy, with 2 planned interim analysis at approximately 35% and 70% of the target total number of cases, which is 151, and with a one-sided O'Brien-Fleming boundary for efficacy and an overall one-sided error rate of 0.025. The efficacy of the vaccine could be demonstrated at either the interim or the primary analysis, performed when the target total number of cases had been observed. The Diemert alpha spending function was used for calculating efficacy boundaries at each analysis. At the first interim analysis on November 15, 2020, vaccine efficacy had been demonstrated in accordance with the pre-specified statistical criteria. The vaccine efficacy estimate, which is based on the total of 95 adjudicated cases, which was 63% of the target total, was 94.5%, with a one-sided p-value of less than 0.001. To reject the null hypothesis that vaccine efficacy would be 30% or less. Now, this is a very sweet p-value. In a future video, I can come back and discuss about all these statistical techniques where I discuss a particular paper that came up with this idea. But for now, we're just going to accept that these are standard statistical techniques to determine the efficacy of the vaccine. Vaccine efficacy was assessed in different subgroups of the population, which included the full analysis population, which is the randomized participants who received at least one dose of the vaccine or placebo, the modified intent to treat population, which is the participants in the full analysis population who had no immunologic or virologic evidence of COVID-19 on the first day or before the first dose, and the per protocol population, which is the participants in the modified intent to treat population who received 2 doses with no major protocol deviations. This vaccine efficacy was defined as the percentage reduction in the hazard ratio for the primary endpoint between the vaccine and the placebo. To meet the regulatory agency's requirement of a median follow-up duration of at least 2 months after completion of the 2-dose regimen, a second analysis was also performed with an efficacy data cutoff date of November 21, 2020. This second analysis is considered the primary analysis of efficacy with a total number of 196 adjudicated COVID-19 cases in the per protocol population. Now, this number exceeds the target total number of cases, 151, as specified in the protocol. This was an increase from the 95 cases observed at the first interim analysis data cutoff date of November 11, 2020. And towards the end, we'll come back and discuss what this exactly implies or what this means. Now let's talk about the results, starting with how the study went, followed by the baseline demographic statistics, then safety data, and lastly, efficacy data. The figure here shows how the trial went. Between July 27th and October 23 in 2020, a total of 30,420 participants underwent the randomization, with 15 to 20 participants in each group assigned to receive 2 doses of either the placebo or the actual vaccine. More than 96% of participants received the second dose, and the common reasons for not receiving the second dose were withdrawal of consent, which was done by about 153 participants, and the detection of the virus itself, done by RT-PCR testing before the administration of the second dose on day 29, which was done by 114 participants, 69 in the placebo group, and 45 in the vaccine group. The primary efficacy and safety analysis were performed in the per protocol population and safety populations, respectively. Of the participants who received the first injection, 14,073 of those in the placebo group and 14,134 in the vaccine group were included in the primary efficacy analysis. 525 participants in the placebo group and 416 in the vaccine group were excluded from the per protocol population, including those who had not received a second dose by the cutoff date of day 29. As of November 25, 2020, the participants had a median follow-up duration of 63 days after the second dose, with 62% of the participants having more than 56 days to follow up for the reaction to the vaccine. On to baseline demographics, which was balanced between the placebo group and the vaccine group. The mean age of the participants was 51.4 years. About 47.3% of the participants were female. 24.8% were 65 years of age or older, and 16.7% were younger than 65 years of age and had predisposing medical conditions that put them at risk for severe COVID-19. The majority of the participants were white, around 80%, and the racial and ethnic proportion were generally representative of the US demographics, including 10.2% Black and 20.5% Hispanic or Latino. Evidence of infection at baseline was present in 2.3% of participants in the vaccine group and in 2.2% in the placebo group, as detected by the RT-PCR testing. Here I am showing you the summarized baseline demographic table one more time. Let's talk about safety statistics. Like mentioned before, safety checks were conducted after the first and second dose for both the vaccine group and the placebo group. This check looks for any adverse event for which there is a reasonable possibility that the test product or the vaccine caused the adverse event. The term solicited adverse reaction refers to selected signs and symptoms occurring after injection administration during a specified post-injection follow-up period, which is the day of injection and 6 subsequent days. The solicited adverse reactions are recorded by the subject in e-diary. The occurrence and intensity of selected signs and symptoms is actively solicited from the subject during a specified post-injection follow-up period using a predefined checklist in the e-diary. The figure on the screen shows the summary for adverse events. Solicited adverse events at the injection site occurred more frequently in the vaccine group than in the placebo group after both the first dose and the second dose. In the vaccine group, injection site events were mainly grade 1 or grade 2 in severity and lasted a mean of 2.6 and 3.2 days after the first and second doses respectively. The most common injection site event was pain after injection. Delayed injection site reactions, those happening after day 8, were noted in 244 participants after the first dose and 68 participants after the second dose. Reactions were categorized by erythema, induration, and tenderness, and they resolved following 4 to 5 days. Solicited systemic adverse events occurred more often in the vaccine group than in the placebo group after both the first dose and the second dose. The severity of the solicited systemic events increased after the second dose in the vaccine group, with an increase in proportions of grade 2 events and grade 3 events. Solicited systemic adverse event in the vaccine group lasted a mean of about 3 days after either the first or second dose. Both solicited injection site and systemic adverse event were common among younger participants, or more common among younger participants than among the older one. Remember, the age categories are 18 to 65 or 18 to under 65 and 65 and older. Solicited adverse events were less common in participants who already had COVID-19 at baseline or who were positive for the infection at baseline than those who were negative at baseline. Let's talk a little bit about some important notes about deaths. 3 deaths occurred in the placebo group. 1 from intra-abdominal perforation, 1 from cardiopulmonary arrest, and 1 from severe systemic inflammatory syndrome in a participant with chronic lymphotic leukemia and diffuse bullous rash, and 2 in the vaccine group as well, 1 from cardiopulmonary arrest and 1 by suicide. The frequency of grade 3 adverse events in the placebo group was very similar to that in the vaccine group, as was the frequencies of medically attended adverse events and serious adverse events in both groups. What about the people who were disqualified from receiving the second vaccine? Overall, 0.5% of participants in the placebo group and 0.3% in the vaccine group had adverse event that resulted in their not receiving the second dose. And less than 0.1% in both groups discontinued participation in the trial because of adverse events after any dose. Adverse events that were deemed by the trial team to be related to the vaccine or placebo were reported among 4.5% of participants in the placebo group and 8.2% in the vaccine group. The most common treatment-related adverse event in placebo group and the vaccine group were fatigue and headache. Now let's talk about the efficacy. What you're looking at on your screen is the investigation of primary endpoint, and this might be one of the most important graphs of this century unless another vaccine cures another pandemic. This can be compared to the efficacy of the BioNTech vaccine as well. However, something like this can probably be considered one of the most important graphs of this century. After day 1 and through November 25, a total of 269 COVID-19 cases were identified with an incidence of 79.7 cases per 1,000 person years among participants in the placebo group with no evidence of previous COVID infection. For primary analysis, 196 cases of COVID-19 were diagnosed, 11 cases in the vaccine group, and 185 cases in the placebo group, indicating a 94.1% efficacy of the vaccine for the prevention of symptomatic COVID infection as compared with the placebo. Findings were similar across key secondary analysis, including assessment starting 14 days after dose 1. An assessment including participants who were COVID seropositive at baseline in the per protocol analysis, indicating a vaccine efficacy of 93.6%. Between days 1 and 42, 7 cases of COVID was identified in the vaccine group as compared with 65 cases in the placebo group. What about the secondary endpoint, which is preventing severe COVID-19? 30 participants in the trial had severe COVID-19, and all 30 were in the placebo group, which indicates a vaccine efficacy of 100%. And that's very, very impressive. And one death among these participants was attributed to COVID-19. The vaccine efficacy to prevent COVID-19 was consistent among subgroups stratified by demographic and baseline characteristics. Among those who were positive for COVID through testing at baseline, one case of COVID that was diagnosed among the placebo, and no cases were diagnosed among the vaccine recipients. Lastly, on to the conclusion and discussion. In this section, we'll summarize overall what we found and discuss some limitations of the study. Let's start with the summary. The COVE trial provides evidence of short-term efficacy of the mRNA-1273 vaccine in preventing symptomatic COVID infection in a diverse adult trial population. Of note, the trial was designed for an infection attack rate of 0.75%, which would have necessitated a follow-up period of 6 months after the 2 vaccine doses to accrue 151 cases in 30,000 participants. The pandemic trajectory accelerated in many U.S. regions in the late summer and fall of 2020, resulting in rapid accrual of 196 cases after a median follow-up of 2 months. It is important to note that all the severe COVID cases were in the placebo group, which suggests that the vaccine is likely to have an effect on preventing severe illness, which is a major cause of healthcare utilization, complications, and death. The finding of fewer occurrences of symptomatic SARS-CoV-2 infection after a single dose of mRNA-1273 is encouraging. However, the trial was not designed to evaluate the efficacy of a single dose, and additional evaluation is needed. The vaccine performed better than vaccines for other respiratory viruses, such as the inactivated influenza vaccine, which has a pooled efficacy of 59%. The efficacy of the mRNA-1273 is in line with the other vaccine from Pfizer called the BioNTech 162b2. Overall, the safety of the mRNA-1273 vaccine regimen and platform is reassuring. No unexpected patterns of concern were identified. The reactogenicity associated with the immunization with mRNA-1273 in this trial is similar to that in the phase 1 data reported previously. Overall, local reactions to vaccines were mild. However, moderate to severe systemic side effects such as fatigue, myalgia, arthralgia, and headache were noted in about 50% of participants in the mRNA-1273 group after the second dose. These side effects were transient, starting about 15 hours after vaccinations and resolving in most participants by the second day. Another positive event is that the mRNA-1273 vaccine did not show evidence in the short term of enhanced respiratory disease after infection, a concern that emerged from animal models used in evaluating some SARS and Middle East Respiratory Syndrome, MERS, vaccine constructs. Let's talk a little bit about some key limitations of the vaccine. The primary one being the short duration of safety and efficacy follow-up. The first point, this high apparent efficacy of mRNA-1273 is based on short-term data, and waning of efficacy over time has been demonstrated with other vaccines. The second limitation, a risk of acute hypersensitivity is sometimes observed with vaccines. However, no such risk was evident in COVE trial, although the ability to detect rare events is limited given the trial sample size. The anecdotal finding of a slight excess of Boyle's palsy in this trial and in the BNT162b2 vaccine indicates concern that it may be more than a chance event, and the possibility bears close monitoring. Next limitation, whether the mRNA-1273 vaccine results in enhanced disease on exposure to the virus in the long term is unknown. Another limitation is the lack of an identified correlate of protection, a critical tool for future bridging studies. As of the data cutoff, 11 cases of COVID-19 had occurred in the vaccine group, a finding that limits our ability to detect a correlate of protection. As cases accrue and immunity wanes, it may be possible to determine all the possible correlates. Although the trial showed that mRNA-1273 reduces the incidence of symptomatic SARS-CoV-2 infection, the data were not sufficient to assess asymptomatic infection. And although the results from a preliminary exploratory analysis suggest that some degree of protection may be afforded after the first dose, evaluation of the incidence of asymptomatic or subclinical infection and viral setting after infection are still underway to assess whether vaccination affects infectiousness. The relatively smaller numbers of cases that occurred in older adults and in participants from ethnic or racial minorities, and the small number of previously infected persons who received the vaccine limit efficacy evaluations in these groups. And lastly, pregnant women and children were excluded from this trial, and additional evaluation is needed to determine the efficacy of the vaccines in these subgroups. So essentially what they're saying is that it's good for everything, almost everything that they try to study, but cannot know everything in the long run. So long story short, within 1 year after emergence of this novel infection that caused the pandemic, a pathogen was determined, vaccine targets were identified, vaccine constructs were created, manufacturing to scale was developed, phase 1 through phase 3 testing was conducted, and data have been reported. The process demonstrates what is possible in the context of motivated collaboration among key sectors of society, including academia, government, industry, regulators, and the larger community. Lessons learned from this endeavor should allow us to prepare better for the next pandemic pathogen. Again, I believe this is a marvel of science, but there are information about the impact of vaccines in the long run that is yet to be known. I would say so far, the evidence overwhelmingly suggests benefits is higher than the cost. Now, this was just a brief summary. Please read the full paper and multiple attached appendix and protocol documents to understand all the nuances of the study. I can do another video if people are interested in the details. The video will be hours, if not days long, and you can probably see that just based on the number of people involved in the study, which is a proxy for the amount of work done. Lastly, before I wrap up, the usual disclaimer. The video is for information purposes only. This is not a legal advice or advice in any professional capacity. I am not affiliated with the authors or the organization producing research or the journal itself. This is my interpretation of the paper. Some statements are direct quotes from the paper itself. Depending on the context, there may be laws around necessary licenses or training or permissions required to conduct the research. There's also always more to a research than what's written on the published paper. I am only trying to interpret or extract what's provided in the published paper. There may be nuances behind this research that may not be explicit while reading the published version. Things do get cut and edited in the peer review process. Caution is advised. The copyright to the paper is owned by the publisher and or the authors. Please reach out to them for more information. I will put a link to the original paper in the description. I hope you enjoyed it. I will be back with another fun and informative paper in the next video. Please do like, subscribe, and comment to let me know if you liked it or you want something changed. Let me know if you want more papers about vaccines. Until then, see you in the next video.